Welcome to the deep dive. Today we're digging into the body's immune system, specifically that first super fast response. But we're starting maybe somewhere unexpected. There's this link apparently between obesity, visceral fat, and how well this whole system works.
Yeah, it's a really important modern angle on, well, an ancient defense mechanism.
Yeah.
I mean, the body's ability to fight off disease hinges entirely on recognizing what's foreign or dangerous. So we're diving into what's called innate host resistance. It's this preset system, always ready. It's your first line of defense, the one keeping you going in those critical first hours of an infection.
Okay, right. So let's map it out. Immunity. It's basically two systems working together, yeah.
Yeah.
You've got innate immunity, always on, hits everything hard, but doesn't really learn. And then the adaptive system. That one needs activation, but it gets specific, and it remembers. So our mission today is to unpack that innate shield. Oh, and quick definition, anything the immune system spots, foreign or even damaged self stuff, we call that an antigen.
Exactly. And you have to start with the physical barriers. The sources really emphasize this. Physical, mechanical, chemical walls. That's the absolute first line. Stop things getting in.
Okay, the surface. Skin seems like the obvious starting point. It feels tough, but what's the specific mechanism that makes it such a good barrier?
It's clever layering, really. You've got these stratified keratinocytes. They're cornified, tough, and constantly shedding, so it's hard to get through. And importantly, it's already crowded. There's a whole ecosystem of friendly microbes living there, basically taking up all the space so pathogens can't easily set up shop.
Right, so if something does get past the skin, it hits the internal surfaces, the mucous membranes, like in your gut, lungs.
Exactly. The GI tract, respiratory, genitourinary tracts, they all rely on this sticky trap called mucus.
Mucus.
Yeah, it's this fluid rich in glycoproteins. It resists penetration, traps invaders. And lining those membranes, you find specialized cells like Paneth cells, even like little defense posts. They secrete things like lysozyme and these antimicrobial peptides right under the surface.
Lysozyme, yeah. And the respiratory system has that cool mechanism, the mucociliary escalator.
It's brilliant. Basically a self-cleaning conveyor belt. Tiny hairs, cilia, they beat upwards, sweeping mucus and anything trapped in it towards your mouth. You cough it out or swallow it. And if a microbe somehow gets past that, way down into the alveoli and the lungs, then it meets the resident guards. Alveolar macrophages.
Okay, moving down to the GI tract, that sounds more like chemical warfare down there.
Oh, definitely. The stomach acid is incredibly harsh, pH 2-3. Kills most things outright. Then you've got peristalsis, the muscle contractions moving things along, plus bile, digestive enzymes, the constant shedding of the gut lining. It's a purge system.
And the genitourinary tract, similar idea.
Sort of. It relies a lot on just, well, flushing things out with urination. And the urine itself is acidic, which helps. Anatomy matters too, you know. The female urethra is shorter, which is partly why UTIs are more common. Plus, in the vagina, you have beneficial lactobacillus species. They produce lactic acid, making the environment really acidic and hostile to many pathogens.
Okay, so that's the physical structure. Now moving inside to the chemical defenses, the soluble stuff. You mentioned limiting nutrients first. How does the body kind of starve out invaders?
Right. Well, many bacteria absolutely need iron to grow fast. So our body has these proteins that are like iron magnets. There's lactoferrin. You find it in mucus and breast milk, and transferrin in the blood. They basically grab onto any free iron very tightly, making it unavailable for microbes. It really slows them down.
Clever. And then there are the direct chemical hits. You mentioned lysozyme earlier. That one attacks bacterial walls, doesn't it?
That's
Especially certain types.
That's right. Yeah, lysozyme is a muramidase. It breaks down peptidoglycan, that mesh structure in bacterial cell walls. It's particularly effective against gram-positive bacteria. Makes them just pop open, lysin.
And then we have these other ancient defenses, antimicrobial peptides. Sounds like tiny chemical disruptors.
That's a great way to put it. They're usually small, positively charged, and amphipathic, meaning they have bits that like fat and bits that like water. They basically poke holes in microbial membranes, causing them to leak and die quickly. Key examples are defensins and cathelicidins. They're like a rapid response force. They're even before the main immune cells show up.
Okay, now this is where, for me, it gets really complex but fascinating. The complement system, this huge cascade of enzymes in the blood. What does it actually do?
It's complex, yeah, but it's all about strategic amplification. It has three main outcomes, three big jobs. First, it fuels inflammation. It generates these potent signal molecules, C3a and C5a, that basically shout for help, recruiting immune cells. Second, it can directly kill cells by building something called the membrane attack complex, the MAC. It literally forms a pore, a hole, right through the target cell's membrane.
Wow.
Wow.
Okay. And the third job, you said it helps the cellular guys.
Right, that's opsonization. Complement fragments, especially one called C3b, coat the surface of the microbe. Imagine a bacterium swimming around opsonization is like sticking a giant "Eat Me" sign on it. Phagocytic cells recognize the C3b coating and gobble up the microbe much, much faster.
So these three pathways to activate it, alternative, lectin, classical, they sound complicated, but I guess the point is they all end up doing those three things, cleaving C3 and C5.
Exactly, they all converge. The alternative pathway is often the very first to react. It gets triggered just by bumping into certain repetitive structures on bacteria, like LPS. The lectin pathway uses these host proteins, like mannose-binding protein, or MDP, that recognize specific sugars on microbial surfaces. And the classical pathway, that one usually needs antibodies already stuck to the pathogen, so it kind of links innate and adaptive immunity.
Okay, this whole system needs communication, right? That's where cytokines come in, the language of the immune system.
Precisely. They're small soluble proteins, but they coordinate everything. We group them by function, loosely. Chemokines are like traffic signals telling cells where to go. Yeah. Interleukins mostly act between leukocytes, white blood cells. Interferons are crucial for fighting viruses. And TNFs, tumor necrosis factors, are major drivers of inflammation.
And there are also acute-phase proteins, things the liver pumps out quickly.
Yeah, like C-reactive protein, CRP, and that mannose-binding protein, MBP again. The liver releases them in response to inflammation signals. They bind to microbes, act as opsonines, remember the eat me sign. And they can also help activate complement. It's like an emergency chemical deployment.
Right. So we've covered barriers, chemicals. Now let's talk about the cellular army, the leukocytes, white blood cells, all coming from stem cells in the bone marrow.
Yep, these are the specialized soldiers.
Let's start with the granulocytes. We hear about neutrophils all the time. They seem key.
They are the absolute frontline troops, the shock troops, highly phagocytic, incredibly fast responders, but they burn out quickly, very short lifespan, almost like they're designed to be sacrificial. Then you have eosinophils. They're more specialized, good at tackling larger parasites like worms or protozoa. They release these really caustic proteins. And basophils and their tissue cousins, mast cells, they're loaded with histamine and other vasoactive stuff, key players in allergies and inflammation.
Okay, then the next big group, monocytes floating in the blood, which then become macrophages in the tissues.
Exactly. Monocytes circulate, but when they enter tissues, they mature into these larger, long-lived macrophages. They are the sentinels, the resident guards. They sound the alarm by releasing cytokines, and they are major phagocytes, constantly cleaning up debris and pathogens.
And then the dendritic cells, or DCs, you said they're the bridge. Macrophages sound the alarm, but DCs do something else specific.
Their main job isn't just killing, it's informing the adaptive immune system. They have these long arms, projections to handle their surroundings. They phagocytose pathogens, yes. But then critically, they chop up the pathogen proteins and present little pieces antigens on their surface. They travel to lymph nodes and show these antigens to T lymphocytes. That's the crucial link. They deliver the intel.
Got it. And we can't forget natural killer cells, NK cells. They come from lymphoid cells but act innately. They target our own cells.
Yeah, they're like the internal security force. NK cells constantly check other host cells. They look for a balance of kill and don't kill signals. If a host cell is stressed, infected with a virus, or becoming cancerous, it often loses the don't kill signals. That tips the balance. The NK cell then releases perforin, which makes pores and granzymes, enzymes that go through the pores and trigger the target cell to undergo programmed cell death apoptosis.
It's just amazing how organized it all is. Like the secondary lymphoid tissues, the lymph nodes, spleen, they're not random, are they? They're placed strategically.
Absolutely not random. T cells mature in the thymus, B cells in the bone marrow, those are primary organs. But the secondary tissues are where the action happens. The spleen filters blood, looking for trouble. Lymph nodes filter the lymph fluid draining from tissues. Nodes are packed with macrophages, DCs, B cells, T cells. They're like monitoring stations where those antigen-carrying DCs from infection sites end up.
And in places with high exposure, like the gut, you have specialized zones, MALT, mucosal-associated lymphoid tissue, something about pairs, patches, and M cells.
Right. MALT is crucial. In the gut, ALT, you have pairs patches, which are like lymph nodes embedded in the gut wall. And these specialized M cells act like little gates. They actively sample stuff from the gut lumen, grab antigens, and pass them directly to the immune cells waiting just underneath. It allows for really rapid responses to anything trying to invade via the gut.
Okay, so we have the cells, the locations, but the big question, how do these phagocytes, macrophages, neutrophils, DCs, actually recognize the bad guys? How do they know what's non-self?
That's the genius of the innate system. It doesn't need to recognize every specific microbe. Instead, it recognizes broad categories of molecules that are common to microbes, but not us. Think of it like recognizing a uniform, not a face. They use pattern recognition receptors, PRRs. These bind to two main types of danger signals: microbe-associated molecular patterns, AMPs, things like LPS from bacteria or viral RNA, and also damage-associated molecular patterns, DAMPs, which are molecules released from our own cells when they're stressed or damaged.
Can we break down the main types of those PRRs? The source mentioned Toll-like receptors, NOD-like.
Sure, think of them as different alarm systems for different locations. TLRs, Toll-like receptors, are often on the cell surface or inside endosomes like vesicles. They're like perimeter sensors. When a TLR binds its MMP, it triggers signaling pathways inside the cell, often activating transcription factors like NFPA, which basically tells the cell nucleus, start making inflammatory cytokines. Now,
Okay, so TLRs are for outside or just inside vesicles. What if the invader is actually loose inside the cytoplasm?
then you need the internal alarms. That's where NLRs, or NOD-like receptors, come in. They float around in the cytoplasm, sensing intracellular MAMPs or DAMPs. If NLRs detect danger, they can cluster together to form this large protein complex called the inflammasome. The inflammasome is really important because it activates some potent inflammatory cytokines, especially IL-1 and IL-18. And then there are RLRs, Rig-I-like receptors. These are also in the cytoplasm, specifically evolved to detect viral RNA. Finding viral RNA loose in the cytoplasm is a major red flag, so RLRs trigger a strong antiviral response, including interferon production.
Okay, alarm sounds, recognition happens, then the killing. You mentioned phagocytosis for external things and autophagy for internal. Both end up merging with a lysosome.
Exactly. Phagocytosis is engulfing something large from outside into a vesicle called a phagosome. Autophagy is like the cell using a membrane bubble to capture stuff already inside, forming an autophagosome. Either way, that bubble then fuses with the lysosome, which is full of digestive enzymes, creating the phagolysosome or autolysosome. That's the killing chamber.
And inside that chamber, it gets nasty, the respiratory burst.
Oh, yeah. When phagocytes get activated, an enzyme called NADPH oxidase switches on. It uses oxygen to generate really toxic molecules called reactive oxygen species, ROS, things like superoxide radicals, hydrogen peroxide. It can also lead to reactive nitrogen species, RNS, like nitric oxide. These ROS and RNS are incredibly damaging to microbes. It's like flooding the chamber with bleach and acid.
Wow. But even this destruction serves another purpose, right? It feeds back into the communication.
Absolutely. After the microbe is killed and digested in the phagolysosome, the phagocyte, especially macrophages and DCs, take some of the leftover fragments and loads them onto those MHC proteins we mentioned earlier, presenting them on the cell surface. It's antigen presentation. It tells the adaptive immune system, this is what the enemy looked like. Get ready.
Which brings us full circle, really, to the integrated response that pulls all this together. Inflammation. We tend to think of it as bad pain, swelling, but acute inflammation is vital.
It's absolutely essential. It's the body's coordinated, local response to injury or infection. Those 5 classic signs: redness, warmth, pain, swelling, and loss of function, they all happen for a reason. They're signs the defense is working.
Let's just touch on that recruitment process again. How do those neutrophils actually get out of the blood vessels? It sounds tricky.
It's a beautiful cascade, actually.
Okay.
First, damaged cells at the site release chemokines, those come-here signals. Then the cells lining the blood vessel, the endothelium, start displaying sticky molecules called selectins. Neutrophils rushing past kind of lightly grab onto these, which makes them slow down and start rolling along the vessel wall. That's called margination. As they roll, other adhesion molecules on the neutrophils, called integrins, get activated and bind really tightly to the vessel wall. This stops them completely. Then, they actively squeeze themselves between the endothelial cells and out into the tissue. That's diapedesis or extravasation. It allows them to follow the chemokine trail right to the source of the problem.
Amazing. And the chemicals causing the swelling and pain.
A key player is bradykinin. It gets generated during inflammation, and it does 2 main things. It makes the capillaries leaky, leading to swelling or edema, and it stimulates nerve endings, causing pain. Bradykinin also prods mast cells to release histamine, which makes the capillaries even leakier, so you get this amplification loop driving the local swelling.
Okay, so acute inflammation is good, necessary, but this system is powerful, almost destructive. If it doesn't switch off, that leads to chronic inflammation.
Exactly. Acute inflammation should resolve, usually within days or weeks. But if the stimulus persists, maybe an infection the body can't clear, or constant irritation, you get chronic inflammation. And that is damaging. It's characterized by a persistent influx of immune cells, like lymphocytes and macrophages, ongoing tissue damage, and often scarring or fibrosis. Sometimes, if the body really can't eliminate something persistent, like the bacteria causing tuberculosis, it tries to wall it off by forming a granuloma. That's basically a ball of immune cells, macrophages, T cells, surrounded by connective tissue, trying to contain the threat.
And this brings us right back to the beginning, the obesity link.
Precisely. The sources highlight that excess visceral fat, the fat around your organs, isn't just passive storage. It actively pumps out low levels of inflammatory cytokines, driving a state of constant low-grade, systemic chronic inflammation throughout the body. So this fundamental defense system gets tied directly into conditions like metabolic syndrome, heart disease, etc., through this chronic inflammation link.
Okay, so wrapping up the key takeaways, innate immunity is all about speed, using preset barriers and chemical weapons. It uses complement for massive amplification. It recognizes general danger patterns, MAMPs and DAMPs with PRRs. And it employs really potent killing mechanisms like phagocytosis and the respiratory burst. And crucially, it's constantly talking to and informing the adaptive system.
That sums it up well. But reflecting on all this, it brings up a final, maybe provocative thought. We've talked about all this destructive power, ROS, RNS, inflammation, all geared towards attack. So why then would the body evolve receptors, like TLR10 is one example the research points to, that seem to do the opposite? TLR10 appears to actively suppress inflammatory responses, to dampen down monocyte and macrophage activity.
Huh. So why build this massive alarm system and then install a dimmer switch or even an off switch?
It really makes you think, doesn't it? It suggests that maybe the most critical part of this incredibly powerful innate system isn't just its ability to turn on and kill, but its ability to turn off. Because if that destructive force is left unchecked, running constantly at full blast, the host itself could be severely damaged by its own defenses. Regulation, turning the volume down, might be just as important for survival as turning it up.
A powerful reminder that control is just as vital as the response itself. Defense without regulation is dangerous. Well, thank you for joining us on this deep dive into the fascinating world of innate immunity.